Hello?
Maria?
Maybe
Yes.
Martin.
Hi, Maria. This is Chris, Lucius, and Mara at UM.
Okay, for people who can hear me, can you please, uh, so I hope we have Kai at this point on the call. Is that correct? Avi,
I'm here.
um, anybody else from your group, Avi?
Yeah, I think there's a bunch of people.
Okay, great. Uh, Kai, we don't, or somebody from University of Washington there?
Yes, I was invited by Kai. This is Mustafa from UKent.
Okay. Nice to So nobody else from University of Washington so far. How about Peter from Cell Signaling or anybody else from Cell Signaling? How about anybody from Miami, Omar or Dushita?
Yes, we are here. Chris is here, Amar and Dushita.
Right. So since the purpose of the conference call is primarily to talk to our external data science research people, will wait a little bit until somebody from University of Michigan and Cell Signaling chimes in.
Siva from Cincinnati also joined. Mario, how are you?
Hi, Siva. It just occurred to me, since we have 2 days conference, that Kai may have decided to call in tomorrow.
Yes, I think she said so in the email.
You did? Okay. Uh, So, Avi, it seems that all 3 of our external data science research groups have decided to call in tomorrow, which is not how I planned it. What do you suggest? Should we each give like short presentations so that we exchange information at least? Because Amar has a presentation, then you guys have a presentation. I can give a little brief introduction so we go through these motions.
Yeah, I think it's a good idea.
Okay. So, all right, I will give a brief intro that was mostly aimed at people who have not been able to access data yet. So I will speed this up a little bit because most of the people on the call actually. Yeah? And I will also, okay, all right, I will not do that then. Maybe just a brief introduction for Mustafa. So Mustafa, what do you plan to access data, analyze data? What is your objective here?
Actually, I'm not really sure yet for now because I was invited by Martin de Cook, which is in the lab with Kayi, I think, at the Washington Tacoma. But I have no previous experience with such data, so this is actually the first time I am involved. But Martin asked me to look into it and see what I can do with it.
Okay, so let me go through my slides fairly quickly, and then this will give you a little bit of information on what the data is all about. And then
Yeah.
I will turn over presentation to either Amar or Avi
Chanan.
Janan. Oh, okay, yeah. I'm sorry. I have him here, so I can pass it to him as well. So that should be a good test for this infrastructure. All right. So the data, basically the L1000 data is meant to profile a whole bunch of perturbation transcriptional signatures at low cost, and it uses the well-established clustering or similarities in gene expression patterns between sets of genes across a large number of samples for this purpose. So the idea is to basically use representative genes from different clusters as to measure their expression at a much reduced cost, and then recreate the whole genome expression profile using a model for the genes that are whose expression is not measured. So this is the idea. Only landmark genes are actually profiled, or their expression is measured. Then using a computational inference model, the expression profiles for other genes or expression levels for other genes are produced. And Broad Institute performed these preliminary studies in which they concluded that 1,000 genes is actually enough to recapitulate most of the information in terms of transcriptional profiles of different perturbations. So the reason for doing this is here. It costs about $5 to generate 1,000 measurements using this Luminex assay, whereas it cost at that point when they did the calculation about $500 to produce a Affymetrix based measurement or profile. And also this Luminex assay is high throughput, so they can generate many more of these. This is the overview of the planned perturbations. So there will be, the plan was to overexpress 3,000 genes, knock down around 3,000 genes with at least 3 shRNAs per gene, and then also chemical perturbations with 4,000 small molecules. And the process of turning this data into measurements is somewhat complicated by the nature of this assay. I'm not going to get into it, but the process goes basically by processing raw data, pair, everything is done at the 384 plate levels. So the data is processed, normalized. After that, the inference engine, so at this point we have measurements for 1,000 genes. Then the inference engine is used to infer expression levels for rest of the 20,000 genes. After that, the ZMAT scores are calculated, which are z-scores per gene. These are calculated by basically subtracting. Again, it's done per plate. So the median expression of a gene is subtracted from each observation, and then divided by the MAD estimator of standard deviation. So this is some kind of somewhat robust z-score. So this is the final, basically, measurement or score of gene expression level per plate that is obtained. And once, so each plate is replicated at least 3 times, and then the z-scores are combined using this moderated z philosophy, in which each z-score is weighted by the pairwise correlation or average pairwise correlation for this replicate with all other replicates, right? So this kind of weighs down the replicates that are not globally correlated with all the other replicates. And this measurement is now called mod-z and represents a summary for each perturbation for which replicates were performed. All right? And this is the level of data that we can now actually, that can be downloaded at this point from the Lynx cloud. So I'm sorry, I'll take this back. This is the final data that is actually not available at this point from the Lynx cloud. The data that is available from Lynx cloud are these z-scores, ZMAT scores, right? So for each replicate, where each replicate has its profile. So the data actually comes in form of this GCTX file. It is a table, compressed table of 22,000 rows and 1.3 million columns. And in order to do any analysis, this data needs to first be parsed. In order to parse this data, one needs to use some of the scripts or software provided in a GitHub repository for L1000 tools, and I will kind of go over this very briefly, how this can actually be done. So this kind of wraps up the just quick overview of what this data is all about. And now my plan was to go through to Lynx Cloud and show the data that where the data can be downloaded from. So the key step is finding two basically sources of data. One is the actual measurements in this big matrix, GCTX matrix, that actually has the z-scores for each perturbation replicate for all genes. And the other is actually to find metadata that describes how all the experimental conditions for each of 1.3 million columns in this matrix. The APIs and the S3, so the matrix is downloadable from Amazon Cloud, and I'm not gonna go into it how to download the data. There are clear instructions. Once you're registered, you can download the data. So we can assume that we actually have the data set. However, I'm gonna just talk about the data that is available. So, look now where this was. Okay. Here we go. So the data that can be downloaded from the Amazon in forms of basically raw or measurements for these 1.3 million experiments, there are 4 levels, and the level 4 are these scores that I was talking about, right? This is the big matrix that that can be parsed to extract the ZMAP scores. You can also get lower processing level data, but that then gets complicated because you have to make some choices about how to derive final scores, and it's probably best to start with the level 4 scores. The metadata can be obtained through this API interface. And so the information that's available there, and one of the, so there are several APIs that allows you to access metadata on cell lines, on genes, profiles, perturbations, plates, signatures, and finally, the one that we are most interested in is this instant info metadata, which basically gives you information about each column in this huge matrix of 1.3 million columns. And in order to parse or extract data from this big matrix, which cannot be processed, and so there is probably, it's probably not possible to load this whole matrix at once, regardless of how much memory you have. And so you basically need to parse it in chunks. And in order to be able to parse it, you need to have identifiers of columns that you want to extract from this matrix. And this is given in as part of the information that you can get by querying this InstInfo using this InstInfo API. And I will not again go into specifics of how do you retrieve the information from there. There are instructions, and it's not completely trivial, but there is it's doable. And again, I'll come back to that a bit later in terms of what you need. But the key identifier that you need is this distill ID, which is basically has the identifier that corresponds to the column names in this big matrix, and you can use these LIDs to parse the matrix. All right. So in order to parse the matrix, you will need tools that are available in the GitHub repository. So again, if you go back to the Linux Cloud and select this for developers link, and then follow the code to code@linkscloud.org. From here, we can actually access GitHub that has all the scripts and sources for processing raw data and for parsing GCDX matrix. So I tried a couple of ways to parse this matrix. I tried MATLAB scripts and R scripts. And I'm going to show you briefly how to use it using R script. but MATLAB is very similar in this term. So now I'm gonna open basically. Let's see. I'll first go to this. So what we have here. So I assume that, assuming that you have downloaded this huge matrix, that's the matrix that we download from Amazon Cloud, 1.3 million columns by 22,000 rows. And okay, so I'm being a little bit advanced. So I'm going to switch back to my R code. So this is the snippet of the R code that you can use basically to parse this matrix. You will need to source a script io.r, which is part of this GitHub repository. Once you download this whole code, this is one of the scripts, and it's this is the location. What I assume here is that you have basically parsed this info stuff from that you obtained from this API that I showed you before, and so that you basically have already created this table, metadata table, that has these still IDs along with perturbation type and cell IDs, which are all information fields that you download from using this info API. If you have that available, then you can parse this matrix by basically specifying the location of your file and columns that you want to extract. And I forgot to remove this. You don't need actually to specify which rows you're going to extract. By default, it's going to extract all the rows in the matrix. So what this is going to do is going to take first 50,000 distill IDs, or first 50,000 identifiers that I obtained from with the API, and extract this object, S4 object, from the GCTX matrix that has these 50,000 columns, the matrix of 22,000 by 52,000 columns. And to access the slot with the data matrix, you just look at the name of the slot is MAT. So these dimensions of this table is 22,000 rows by 50,000 columns, right? Now, these still IDs or the column names in this matrix have a specific format, and you can find some information about how this profile was generating by parsing this name. So the first part is the plate identifier, the second is cell line, then is the time point, then it's the replicates. So X1 is the first replicate, X2, X3, X4, and so on are subsequent replicates of the same plate. There are bead batch and the bead batch set, which are important because the beads actually, the landmark genes change slightly from one bead set to the other. And it turns out that batches in which things are performed also introduce some additional noise, so you may want to process data by batch. Finally, in order to know how big of you cannot, in R at least, this chunk cannot be too big, how many columns you can parse at the same time, and that's determined by your machine precision to some extent, by the maximum integer that it can be represented in your machine. And this is command that gives you how many columns you can download at a time. So the maximum number on my machine is 96,130. So this is an example of how you could use R to parse this, assuming that you download this metadata information using Info API. You can do this in a more interactive way by basically selecting through APIs just the perturbations that you want to use and just the cell lines that you want to parse instead of just downloading the whole info data. But that gets a little complicated, and I guess we can talk about this later. And that would be basically the end of my presentation. So I wanted to be just very focused in terms of explaining how you can go about downloading and parsing this data. Now, I guess we can go either to the Miami group and have Amar tell us about their set of signatures that they have available, or go to Shannon in Avi's group to tell us about an alternative way to process signatures and create this final representation that actually is not available at the moment through the broad cloud.
Maybe before that, we should take some questions.
Yes, question.
So I have a question, if I may.
Please.
Yeah.
So this data is actually open for public access, or are they restricted? And are there any conditions on how whether you can use the data research?
If you go to linkscloud.org, okay, in the follow-up links to download the data, they will explain to you in which what is a fair use policy at this point. So you do need to request access, and
Right.
they will grant you access, and you can use it for non-profit research work.
Right.
In any way you want. That's the general policy, but it's pretty clearly spelled out, basically, under which conditions you can use it.
Right. Right. Thank you.
Does that answer it?
Right. I guess so. Yes. So basically, you can use it, but there is a fair use policy that we have to abide.
Right. So if they will grant you access to data, you're not supposed to share it.
Right.
So it's just access for you to use data in your research, non-profit research.
Right. Right. Thank you.
Any other questions? Okay, so who would like to go next?
Great. Amar would like to go.
Amar would like to go. Okay. So let me find Amar. Amar, did you identify yourself? Because I need to switch the presentation to you.
Okay, we're downloading the app really quickly.
Okay.
Yeah. It's going to start any second.
Well, all you need to do is find yourself on the list of anonymous viewers and modify so that I can identify who you are, and then I will switch it from my app to you to be the presenter.
Okay.
and you can figure that out because you are able to modify
Okay.
only your ID.
Yep. We have modified it.
You have modified it? Okay. Here, I see you. All right, so I'm gonna pass presenter role to you, Amar. Okay?
Yeah, thank you.
Can you see our screen? Yeah, I guess.
Yeah, we would like to present how we are processing the links data. I mean, the L1000 data and putting into LifeWorks so that it's searchable with other essays. And so this is how we do it. So next.
Do you see the screen?
Yeah, we can see.
Okay.
Index L1000 signatures with the metadata and also the z-scores, which is aggregated z-scores, which is right now not available from the API. So we just want to go over the entire process, and we also want to see if there is any other alternatives than what we are doing right now. So so we take the signatures and yeah. Okay, cool. So this is how the process we have. So the broad gives us the data in two different ways. One from the API and one from the S3 server. So S3 server, what we do is we just download the entire GCTX file, which has all the signatures in it. It's not the signatures, it's the distill IDs and the Z-scores that they have. And then what we do is we take the GCTX file, parse the entire GCTX file and cache it into a database. And then what we do is, based on the distill IDs that we get from the TX file, we send the distill IDs again to the API and get the signature ID. Okay, and then we store the signature ID and the combination of signature ID and the distill ID into another the table. And when we query the broad API, we also get metadata info like the cell line the signature is associated with, and concentration, time point, all those information, and then put it into a database. And at this point, what we have is a signature ID, and then it has multiple replicates, which are distill IDs. But we don't have an aggregate at this point. So what we do is we aggregate that in our end. So the aggregation is the simple aggregation. The aggregation is a simple method that we use right now, which is like we take for a given distill ID, for a given signature, we take the average of the gene Z-score for a given signature. So that's how we do it, and then we create indexes. So I'll just go in detail about these. So as you know, there are four data levels that Cloud provides: raw data, processed, and normalized data, and then aggregated. Aggregated is the Z-score. So
So,
Right now,
Mark,
right?
can I interrupt you just for a second?
Yeah, sure.
So this is not the case at the moment, right? Just wanted to make clear. At the moment, the data, level 4 data are not aggregate signatures.
Yes, that's what we do here. I mean, we aggregate those Z-scores. It's not
Okay, I
available.
got you. So you actually aggregate them. All right. So your
Yeah.
level 4 is different from level 4.
Yeah.
Okay.
So, I mean, we associate, so the signature ID that they give you, that they give us, is an aggregate of all the distill IDs, right? Right?
Right.
The signature, but the Z-scores are not aggregate.
Right.
Right? So what we do is we aggregate the Z-scores and add it to the signature IDs that they provide. So it makes, right now what Broad gives us is the signature IDs, which is aggregate, but they don't provide aggregate Z-scores. And we calculate ourselves those aggregate Z-scores. That's what I want to say.
So you just average, for
Yeah.
each signature, you average all the Z-scores that are listed as being
Yes,
part
for
of
a given gene. Yeah, for a given gene.
Okay.
And so, as I said, there are two ways. One from the API, we get the metadata for the signature, and then from the S3 server, we get the GCTX file. They're like, so there is Transcenter project as well as Lynx Joint project as well as the Lynx L1000 project. So both of those GCTX files are available from the S3 server. So we just download them, and then we use the Python and the Java processor that the API, the Lynx Cloud provides. So there are some just like you showed us the R package that you were using. We were using the Python and then the Java package. So we extract the distill IDs, Perturbagen Z-scores from the GCTX file and put it into a database. And from the API, as you know, the API is organized into various collections like cell info, gene info, PERT info, signature instant, and plate info. We only take the sig info and the gene info right now because cell info is, so we follow the metadata standards that are imposed by the Lynx group. So for that, which means that we ask the Broad for the smiles and stuff, and then we standardize it ourselves across the various Lynx centers. So that's why we don't collect the PERT info from the Broad. We query the gene info to get all the and the sig info at this point, and we from the sig info we extract we pull a lot of information like the cell ID, signature ID, and all of those things, and then put all of those into a MongoDB. This is where all the signature data is stored in, because the data that is retrieved back from API is in a JSON format, and it makes sense to put it into MongoDB. So we go and we just put it into Mongo, catch it into Mongo. Yeah, this is just one example of how SIGINFO, one result from the SIGINFO. I did not put everything. Just I just want to show the distill ID and the SIG ID, because SIG ID is the signature, and the distill ID is the associate. Those are replicas. So these are the main information that we gather, and then to integrate both the GCTX file and the API, what we do is, as you know, GTCX file is at the digital level, I mean at the replica level, and the API is at the signature level. So in order to put both things together, what we do is we aggregate the Z-scores. Okay? The Z-scores are aggregated on the signature. Right. And we only from the aggregate we take the top 50 up and down regulated genes, and that's what we use it. Yeah, to put into live birth. So in life we don't have all the 22,000 genes or all the 1,000 genes expressed for a gene for a given signature, but we have top 50 up regulated and top 50 down regulated genes. That's what we have in life, like that. And it's the same thing. So when we query the API, we get some metadata, right? Like quarter version and cell line and stuff. So if there are anything new that is not already standardized, we go request and collect the metadata information and standardize those across. Yeah, sorry.
So can I ask you another question? So at the end of the story, you again boil down this to 100 most up and 100 most down regulated genes. Is that correct?
Yes.
For storing things in the.
Yeah, when we expose from life, we only expose the top 50 up and down regulated genes.
But these are yours based on your aggregation.
No.
This code, or these are from the SIGINFO.
Yeah, these are from the SIGINFO.
Okay.
It's not. Yeah, it's coming from the SIGINFO. And we standardize these molecules and then index it to so that it's searchable. So there are a lot of small molecules that overlap between the various link centers. So there are some compounds in HMS and some in broad. We standardize. We take the SMILES and standardize those. So I'll just show you the process of how it is standardized. So we'll take the center provided SMILE, and then desalt the files so that we get the SMILE at the compound level, and then based on that we standardize. So you can see and generate an LSMID, and we also get other information, like whether it's a PDB ligand or whether it's a known drug, or whether it is a kinase inhibitor or those other things from various sources, and annotate that to a compound. So what this helps us to do is something like I just want to show one example where we search L1000 and kinome scan. So the L1000 is for L1000 transcription response assay and kinome scan assay. We want to see all the small molecules that are tested in both, and you can do it with a simple query saying L1000. Just have to hit L1000 and kinome scan to get all the compounds that are in common. And you want to, we also have tags to associate to compounds, so you can use other filtering. Say that only give me compounds that are PDB ligands, and you will see only the PDB ligands. Yes, there are 41 molecules that are shown on the slide. So we have two indexes. One is constituent index and perturbation index. Perturbation index is where you have like the endpoint details for a given signature and stuff. So for a given signature, if there are like 100 genes up upregulated, 100 genes downregulated. So perturbation index will have gene for the signature you'll have like 200 perturbations, because each combination of signature, gene, and z-score is one kind of perturbation for us. So that's what we use. So these are the overall stats that we have right now in live. So total signatures in plot. It shows the total signatures. So these are three different signatures that plot provides, like overexpression, knockdown, and compound. There are many other, but we are only focused on these three, right? So when we only expose the gold signature, we don't expose. At this point, we don't expose all the signatures. There are a lot of signatures that plot has, but they say some of those are very gold, which are better, and then we only expose those gold signatures. So this are the complete stats of what. We are also there's more data cyber set. So there are some cyber compounds that are listed already, and they have L1000 bringing those as well. There are some cyber also, the cyber group also type is yes, some of. And as I said, we use the metadata specification so that data can be searchable across centers. So that's so here is where you can find all the metadata for metadata standards for various types, like various constituents, for cell lines, genes, shRNA, and other things. So we are trying to standardize lines and compounds at this point, but we also want to do proteins, but right now we are working with HMS on that, and we can, yeah. These are all the data standards that are available. Yeah. Thank you. You have any questions? Hello?
A few questions. No more from Mario?
So I think it would be a good. So the bottom line is, you guys do have final signatures that you get by aggregating all the z-scores for each signature by simply averaging them. But you don't expose them on live, but perhaps you would be able to share maybe with external data science research projects if they would like to use them. Would that be a good bottom line? Because now we can go to Xiaonan, who actually has an alternative way to create these signatures. And at the end of the story, Bruce has a third way. As I kind of showed, they use this moderated z-score. So we already have three different versions of signatures that maybe give us different results. And
one thing that we can do, we can benchmark those different ways, and we can actually tell you which one is better. And
yeah,
Xiaonan has some ideas of how to do it. He has that in his presentation, only part of it. But I can, share other ways to do it.
But this is benchmarking is great, but it's kind of a little independent on how or what we serve, right? Or what we, the most important thing is we describe exactly what was done and what your signatures are, right? I'm not saying that the quality doesn't matter, right? But the most important thing is, right, you need to make very clear what exactly was done, so that people don't get confused. For example, if we use another better method, right, and then they compare that method and the broad, they get different results, they could be very confused. We need to make it very clear what's what, right?
Yeah.
Sounds good. So why don't Avi, you think we should go to Chana now? Can he kind of go through his presentation in the remaining time, or?
Yeah, I think he has to be quick. I mean, he has 2 parts, so maybe tomorrow you'll have more chance, depending on what we want to do tomorrow. But you'll have more chance maybe to do a practical demo, and today you can talk more, like just go over his slides.
Okay. So why don't I pass the presenter role? Thanks, Amar, and I'm gonna pass presenter.
So, hello everyone, could you hear me?
Yes.
Yeah, but we can't see your slides yet.
Okay, so here I will briefly talk about.
We can't see your slides. Chana. Oh, you can?
Yeah.
Okay.
We can
Okay. I see it. Sorry.
Yeah, I think others could see. So
I see.
Okay. So basically, it's a method to analyze the links L1000 level 3 normalized gene expression data sets. So it's basically an alternative way to provide signatures for experiments, an alternative level for signatures. So here's an overview of the methods. So the methods have 2 parts. The first part is to identify significant experiments, which are experiments that are significantly different from controls, right? And the second is to identify significant genes, that differentially expressed genes. For the first part, I use only the 978 landmark genes. And for the second part, I use the full gene set, which is 20,000 genes. So the first part is, the result from the first part is good for experimental level analysis, which you do not want to involve any genes, such as clustering analysis. The second part is good for gene level analysis. For example, if you want to do enrichment analysis. So here's the experiment setup. I think Mario pretty much already explained it. So I want to briefly describe it here because it has the consequence on how we analyze the data. So the purple is control, the blue is the experiment. So here is the concept of batch. I think abroad we'll call it a perturbation group. So I just call it a batch. So a batch is a group of experiments that share the same plates. So here is a batch of 3 plates, and for each plate it has control replicates and the experimental replicates. I think the broad arrange the experiment as such. So for example, for one experiment, it will have the same number of replicates as the number of plates. And the replicate of the same experiment will be placed on the same well of each plate. So for example, this is the replicate in plate 1 for the experiment, and this is the second replicate, and this third replicate for the same experiment. Here's some statistics. It's about 80 control replicates per plate, and generally there are 2 to 4 plates per batch. And so generally each experiment will have 2 to 4 replicates, and there will be about 360 experiments per plate because you have to subtract 384 by the number of control replicants. So here we come to the first part, identify significant experiments. Actually, the method I used is an adaptation of the character with direction method our lab published this year. This is a method invented by Neil Clark in our lab. I just tailored this method specifically for the links data. Here is a toy example to briefly explain this method. So now we have a gene expression measurement of 2 genes for experiment and control. Now we want to, first we use the LDA, linear discriminant analysis, to find a boundary that best segregate experiment from control in this 2 gene expression space. And then we will find the direction which point from control to experiment and perpendicular to this boundary as the direction that characterize how the experiment replicates are different from the control. So this vector will be a direction, so it will be a unit vector with a norm of 1. And this will be the single representation of the experiment. So now let's look at the projection of this direction on the axis of these 2 genes. So basically, this direction will be represented as a vector of 2 components. The first component will be the projection on gene 1, and the second projection will be the projection in gene 2. And we can see that the squared value or the absolute value of gene 1 is larger than gene 2. And so which means that the control is more different than the experiment is more different from control on gene 1 than gene 2. So the gene 1 contribute more to the difference between experiment to control, and in other words, that the gene 1 is more differentially expressed than gene 2. And then let's look at the sign of the component value, whether it's positive or negative. So the gene 1 has the positive component, which means the gene 1 is upregulated in experiment, and gene 2 is, as you can see, it's negative projection. So gene 2 is downregulated in experiment. So basically, use one single direction representation. We can use the component value to describe the first, how much a gene is differentially expressed, and the second, whether the gene is upregulated or downregulated. Now we can calculate the direction for other experiment. For example, calculate the direction for the second experiment and the direction for the third experiment. Then we can compare the similarity between experiments by use these directions. So if 2 directions are aligned together, which means the experiment will be more similar. If the 2 directions, the angle between the 2 extensions are large, they are not aligned to the same direction, so they are not similar. And this similarity and dissimilarity could be characterized by this called sin distance, which is basically a matter of the angle between 2 directions. So I will call this to mirror the similarity between experiments. This is a PC principal component analysis plot of of an experiment and its replicates. So these replicates of this experiment has 4 replicates. So these are the controls for the first replicate in plate 1, and these the control replicates of the second plate. These are the control replicates of the third plate. This control of the fourth plate, and these dot are the experiments. So we can see there are systematic difference between the data mirrored on different plates, which certainly doesn't reflect any biology. So we call it the plate effect, and it should be corrected. So the way we correct this effect is to calculate a direction of the experiment on each plate, using the single experiment replicate and the control replicates on this plate.
Can I ask you just a very quick question? So
Okay.
Is it just Q normalized observations?
Oh, yeah. These are quantum normalized data.
Okay, but not z-scores,
So-
not Vmax.
Not the scores. Like we started from level 3, which are normalized data, and we derive our own signatures. Nothing related to this score. So to correct this plate effect, so then we calculate the same number of directions as the number of replicates. So we call this characteristic direction replicates, just CD replicates. So like for this experiment, it has 4 replicates, so it has 4 CD replicates. Then we will basically average all these CD replicates to get the direction for this experiment, just average. Now I will introduce the concept of average cosine distance. These 3 directions are the characteristic direction replicates of one experiment. So this is the direction calculated from plate 1. This is the CD calculated from plate 2, and this CD calculated from plate 3. Remember, we use this cosine distance to measure how much 2 directions are aligned together. So we can compute the cosine distance for each 2 of these 3 CD replicates, and we get 3 distances. So the average cosine distance will be all these 3 distance averaged by 3. So this average cosine distance is basically a representation of how much these CD replicates are aligned together. So if an experiment is not significantly different from control, its CD replicates will be random. Then their CD replicates, these replicated directions will not be aligned together. And since it's a very high dimensional space, 900 or 20,000, so there's a very, very little chance that 2 replicate directions could be aligned together. So the average cosine distance will be large. But if experiment is very significantly different from control, their CD replicates should pretty much align in the same direction. So the average cosine distance will be small. So basically, small average cosine distance, the stronger the experiments, and the larger the average cosine distance, the weak the experiment. So it's basically an estimation of experiment strength. So we benchmarked this average cosine distance by evaluation of the CD method to detect the experiment strength rank. So the basic assumption is that if all the other conditions are the same, the experiment of a higher dose should elicit a stronger response. So I just correlate the dose rank, the experiment ranked by dose, and the experiment strength rank for about 800 sets of experiments. And these experiments in a set are only different by dose. So here's a correlation of using average cosine distance. So it's about 0.6. And here is I use this TOSS, which is a measurement of experimental strength provided in the API level 4 data sets based on these score signatures. So you can see that the average cosine distance is significantly better in the correlation between the dose rank and the experimental strength rank. Okay. But with this average cosine distance, although we can rank experiment from weak to strong, but we want to have a cutoff about to tell us at which point we will define as an experiment as significantly strong and which experiments are not significantly strong. So I use the null distribution to generate the cutoff. To calculate the null distribution, you first calculate all the CD replicates for all the experiments in a batch. So this method is based on batch, so I process batch by batch. So first I compute all the CD replicates for all the experiments in a batch. So this will be the CD replicates of experiment 1, and this batch has 3 plates. The color will suggest the plate. So these are the CD replicates of experiment 1, CD replicates 2, up to CD replicates of 360. Then from this pool of CD replicates, I random draw 3 CD replicates. For example, it might be like CD replicate of experiment 1 from plate 1, and CD replicate of experiment 9 from plate 2, and CD replicate of experiment 181 from plate 2. And then after I will treat these 3 randomly drawn CD replicates as a pseudo experiment and compute its average cosine distance. This will be the null cosine average cosine distance 1. I just repeat this process by 10,000 times to get a null distribution of the average cosine distance. Here's the histogram. Then this is basically the null distribution, and I just draw the upper 5%, which is at 0.9. So this will be the cutoff. So all the experiments which has average cosine distance less than 0.9 will be all the significant experiments, and all the experiments that has an average cosine distance larger than 0.9, they will not be the significant experiment. So after calculate the characteristic directions for each experiment and fill out all those experiments that are not significant, we can do some clustering analysis. Here's show some examples. So this is a multi-dimensional scaling of the character directions of the part of the links data, this RGP04 significant perturbations. And the distance between the experiment are narrowed by cosine distance. So in this part of the links data, they are ligand perturbations to 6 different breast cancer cell lines. The ligands are classed into 4 categories: the growth factors, interference, the interference and this IL interference. So you can say use this characteristic direction. Ligands are separated by their class, but while you use the Z-score signatures, you'll get a cluster like this. So it's pretty much overlapped. You cannot discern patterns. And actually, the character direction can reveal more subtle differences. So here, I colored the same set of dots by time points instead of ligand class. So you can say, like, for some ligands, they are separated by time points. So these are the perturbations at time point 1, and these are perturbations of time point 24. But if you use this cost signatures, it looks pretty much random. You can't see any patterns from this MDS plot.
Can I ask you another question? Just to, we're on the same page about the Z-scores. So these Z-scores are moderated Zs?
Yeah. Like before, like Broad published the moderated Z-score for some part of the data.
Okay, thanks.
So the distance is a mirrored Euclidean distance. So you can say in terms of clustering analysis, the characteristic direction method is pretty much better. So we will come to the second part of identifying significant genes.
Maybe we should do the second part tomorrow.
Okay.
If people have to leave. What do you guys feel?
That would be fine with me. We can post this video where people can access it to see what we talked about today. I'm just, I think tomorrow, if actually we are talking to EDSR people, the emphasis may be slightly different. Their highest priority may be just getting the data and being able to process it versus looking at the fine point. So could you comment maybe tomorrow, or be ready to comment on the availability of these? So would you be able to share these signatures with them so that they can use them?
Shannon?
Yeah. I think I already processed the part of the overall data sets, but it takes time to process all. Like some part of the script takes a long time to run. So, but after I process that, I will definitely sharing the data, sharing the signatures.
Okay, that's great. So yeah, I'm for this to continue tomorrow. I'm just in case that we have, I assume we have more questions tomorrow, and if we don't get to it, the next meeting finish the second part. How's that?
Can you guys post the presentations on the folder that I gave you, that I sent you guys?
Okay, no problem.
Yeah, I mean, it's not public, it's just kind of DCIC folder. And you can add the other folks too. It will be easier maybe for people to look at it.
Okay, sure.
Okay, sounds good. Well, thank you, guys. I think it may be a good time to wrap it up, because if people have to leave. Send me your thoughts. Email me what you think, how this worked out. Is that the proper emphasis, and how we should proceed? Okay?
Okay.
Any final thoughts?
No, thanks for organizing, Mario. Great. I mean, I only dropped in here at the end, but it's great work actually over there. The last stuff. This is pretty cool. Actually, it works much better than the Z-scores.
Yeah, that looks pretty cool. Yeah, I agree.
Because we saw Z-scores ranging from -100 to +100. So I don't know.
Yeah, definitely. There's a little bit of noise in the values, yeah, but the direction is actually kind of very clever.
Yeah. Well, actually, they, from what I understand, these scores are thresholded, right? At -10.
No.
No?
No, not really. No.
No, I mean, this is a threshold quantification. They do the quantile normalization, then they use that to make the Z-scores, right?
Right, but in their QC step, I think they threshold it at -10. Yeah, wrong.
You mean for the is-goals?
No, maybe not, because even on the C3 server they specify that it's -100 to +100.
Okay.
So
All right.
I don't think, because we saw the scores in that range. So.
But I have a few more questions, but I can just ask you guys offline or tomorrow or so.
Well, I think this one thing that I would like to kind of to put additional emphasis, is to kind of get on the same page in terms of what exactly is done by Broad Institute when they generate their Z-scores, so that
I think Amar is a pretty good sense of this, right? Oh, do you?
I don't know how they generated it. I'm not sure how they generated it, but.
Did anybody reproduce this? I mean, like checking basically the quantile normalized data, and when didn't you guys also work on the peak detection, all of that kind of stuff? Anyway, I mean, probably not a discussion for now, but I saw lots of people were working on that.
I mean, I think everybody did a little bit work on that, and we looked at peak detection and all this kind of stuff. But at the end of the story, I think we all settled to using Broad's pre-processing to various extents. I mean, Avi started at Q-normalized data. Amar is using Z-scores, right, to average them. We also basically started Z-scores now. So I think we are kind of on the same page there, that we don't really go back to reprocessing of the raw data.
Yeah. I mean, our main focus was, just rather than trying to maximize the information content, is first make the data searchable in some way. And I mean, that can be done with any method, right? We just need to maybe make a little classification of what the different approaches are, the different similarity measures, right?
Right.
That should be actually would probably make it much more useful. I just wonder. I mean, again, we can discuss maybe these different methods a little bit more some other time.
I think we should, because the for general public, yes, it may be just accessing these things is the primary objective, but for the external data research groups, they will actually want good signatures, and they want to know how they're created probably and what they mean and so on. So.
Yeah, they want to see benchmarks like Avi mentioned and that, right? Because I mean there is. I'm not sure, but I could imagine that there may be different ways of processing them. Perhaps that would be better for different purposes. I'm not sure, but maybe.
Yeah, that's possible.
So if we succeed, right, in doing both of those things, this research of processing and then analyzing the data and benchmarking it, and also like giving like very like kind of intuitive, user-friendly, clear access to it, right, in a way that just makes it pretty clear what happened and what those different things are. I think that would probably be success, you know, in a sense of what we are supposed to do.
Yeah, I really think that would be important. Yes, absolutely. And I don't think there is clarity at this point.
No, it isn't, right? I mean, even among our groups, right? I mean, isn't entirely aligned, you know. So we just have to streamline that, right? And all right.
Okay, cool. So let's see. Hopefully, most of you will be able to show up tomorrow again, and we'll have a different conversation. Okay. Thanks, guys. Bye.
All right. Thanks, guys. Thank you. Bye.
Thank you.
Yes. Okay. Sorry. Yes.